So welcome again, everyone, to today's webinar on merging research on land application and biosolids, featuring Dr. Ian Pepper. And I'm delighted that we have such a great attendance and turnout for this, and I appreciate everyone joining. Before we get down to introducing our featured speakers, I'm going to go over a few housekeeping items. We will have about 20 minutes or so for Q&A at the conclusion of the presentation. So please just type in any questions as we go along using that Q&A function that you should see at the bottom of your screen. And I will mention again, the presentation is being recorded and the recording will be shared out with you. So for those who are looking for that and slides, we will share them no later than tomorrow. So before we do hear from today's featured speaker, Dr. Ian Pepper, I'd like to introduce Chris Peot, who's going to give some introductory remarks, and he will also be part of the Q&A discussion. Again, I am April Thompson, Senior Director for Bloom, and work closely with Chris, who's really the driving force behind Bloom as Director of Resource Recovery for DC Water. Chris directs the recovery of resources at the authority by establishing policies, plans, and procedures related to the recycling and extraction of value from recovered resources, which we're going to hear more about momentarily. Before joining DC Water, he served as a consultant in Seattle, working on organic waste recycling issues and worked on similar issues for LA's Bureau of Sanitation. Chris has a BS in Civil and Environmental Engineering from the University of Wisconsin-Madison and an MS in Civil Engineering from California State University at Long Branch. Chris, over to you.
Hey, thanks. Really appreciate everybody tuning in. As April mentioned, my name is Chris Peot. I'm an engineer here. My title is Director of Resource Recovery. It's our job in my group to maximize the assets that we have, make use of them, return carbon and nutrients back to the earth from which they came, lower our carbon footprints, make green energy, all kinds of different things. The mantra in our group is that there is no such thing as waste, only wasted resources, and we take that very seriously. Our job at the wastewater treatment plant, of course, is to there you go, is to remove the, make sure that carbon and nutrients don't end up in the river and therefore out into the Chesapeake Bay. They're considered pollutants out in the Chesapeake Bay. We don't want them out there because they'll cause algae blooms, premature aging of the Chesapeake Bay, dead zones and all that. So we take that responsibility very, very seriously. We have extremely low nutrient limits and carbon limits on our discharge that goes out into the Potomac River. But those pollutants, carbon and nutrients, are valuable. Carbon is energy and nutrients are fertilizers. So we have put systems in that allow us to extract energy and nutrients and then make use of them. We built and started about 5 or 6 years ago a new digestion system preceded by thermal hydrolysis, which is a European technology. Ours is the first one in North America, the largest one in the world. High heat, high pressure system that ensures that we meet all Class A exceptional quality standards by EPA, killing all the pathogens. The material goes into the digesters. We produce gas out of the top. That gas gets burned in turbines to make, again, clean, green, renewable energy. Turbines spin and they generate a lot of heat. So we recover that heat and then we put it into the thermal hydrolysis system. That's how we heat it up. So it's incredibly energy efficient. But the digesters aren't 100% efficient. They digest what is easily digestible, make gas out of it, and what is less easily digestible or more stable comes out of the bottom. And we squeeze the water out of that, and that is our biosolids product. I manage the biosolids program here, and we make a product called Bloom, and it's regulated. Both the water and the biosolids are regulated for use and designated in order for, and they're designed in order to protect the environment and human health. And again, we take all of that very seriously. There we go. So, the Bloom benefits are that, it's a great value to the farmers, helps helps maintain agriculture in these times when urban sprawl is spreading out into areas around our urban centers. Helps preserve agriculture, improves the soil health, reduces costs for the farmers. It really completes that nutrient and carbon cycle, which we're very, very proud of. Kind of the way the earth is supposed to work. We are part of the ecosystem, and other plants or other animals are kind enough to return their waste to the earth so that we can make more soil. So we're doing that same thing. It dramatically reduces our carbon footprint, protects Chesapeake Bay by not only reducing the amount of nutrients and carbon that go out from our effluent, but also reducing the use of chemical fertilizers. And then it reduces costs for our ratepayers, so it helps keep the rates low for them. You guys see all this? We've got some of it is marked. All right, good. So, we're here today because there are some concerns about PFAS in our society at large, but in particular around biosolids. And it does show up in small quantities in biosolids. I have here a chart here showing how much we've tested for it in our fresh bloom. That's what gets land applied on farms and what gets sent to soil blenders. And the PFOA level is about 3.7. PFOS level is about 15 or 16, and it's pretty consistent. We've measured it a few times, and it's very consistent. But the way it stacks up against other exposures in our society is we're sort of on the low end. Some of the ones that are interesting is household dust and dust at daycare centers is 10 times the concentration. It makes me worry about walking around my house and doing the laundry. I do the laundry at our house, and I take the lint off the dryer filter, and that's one of the ways that PFAS enters the dust in the home. It's in our carpets. We're creatures of convenience, so that when I inevitably spill coffee on my carpet, it's easy to clean. That's why carpeting is saturated with PFAS. So while it is in biosolids, it is at much higher levels in other products that we use. And, the solution is probably to get it out of the products that we use through source control, which we fully support. And recently there were a few states who have addressed this issue in different ways. I want to highlight a couple of them. Maine, facing some backlash from the public after some farms were found to have highly contaminated soils with PFAS. Really did a de facto ban. They set limits that were extremely low that nobody could meet. So all the biosolids had to go out of state. But the sites in Maine, they have thousands of times higher concentration than you find on municipal biosolids treated sites. And that's why Dr. Ian Pepper is here. He's going to go over a study that he did in Arizona and that he is trying to replicate, or is replicating across the nation in different soil types. It's really important to separate PFAS risks and impacts of municipal biosolids such as bloom from the risks presented from industrial sludges from paper mills, for instance. Several of the farms in Maine that had the high levels were treated in the 80s and decades ago with paper mill sludge. Paper is one of the uses of PFAS. It helps resist grease, it helps resist water, and it's heavily used. And again, we're creatures of convenience. And we're finding that these high levels of use of PFAS can have unintended consequences. So we are faced with trying to solve this issue. So in Arizona, there was a situation where, and Ian's going to talk about this in great depth, but I just wanted to do it as a setup, where right around Tucson, Arizona, there were some wells found that contained some PFAS. And they immediately saw that there was land application of biosolids in the vicinity, not right next to the wells, but in the vicinity. And in response, they put a moratorium on the land application of biosolids. And the water authorities said, well, that's not terribly scientific. We don't really know where it's coming from. Let's put some science behind this. And Dr. Pepper did a study that looked at sites that had historic land application of biosolids, did testing on those soils, compared them to soils that didn't have any land application of biosolids, found very similar levels. and then looked at how it migrated through the soil. I'm not going to go into it because Ian's going to do it here shortly, but suffice it to say that based on the findings here, Pima County rescinded that moratorium about 11 months later, and land application of biosolids resumed. So I'm all for studying this. I'm all for regulations. I think it's really important, but I implore regulators to ensure that they are the decisions are science-based. This is one of Ian's graphs that he allowed me to sort of copy for this. It sort of shows the difference between the industrially impacted soils and those that are impacted by municipal biosolids. The three soils in the middle come from the study that Ian did, and the first one, 1.58, is soils that had been land applied for 5 to 10 years. Oops, I just skipped it. The next one was 12 to 20 years, and then the third one on the right, 1.85, were local soils that had no biosolids in them, and they're all about the same level. The one on the right is the Songbird Farm, Maine, one of the farms of interest up there where they had contaminated soils. And it is extremely high, 475 parts per billion. And that we don't have data on the PFAS levels of the paper mill sludges that were land applied there because nobody was testing for it back then. But if you back calculate it, in order to get that level of concentration in the soils, the solids that were land applied would have had to have concentrations of over 100,000 parts per billion. And a typical municipal biosolids like Bloom has about 15 parts per billion. So it's orders of magnitude higher. And it's important to make that distinction. Michigan did make that distinction. They looked at, they surveyed all the wastewater treatment plants in the state, or many of them, and looked at levels of PFAS in the biosolids. And they made a distinction between those that are industrially impacted, those are the ones on the right, the purple ones, and the ones that were largely municipal biosolids. And this looks like the levels are kind of similar, but I just want to remind everybody that this is a logarithmic graph. So the ones on the right are orders of magnitude, 2-3 orders of magnitude higher than the orange ones. And so Michigan then set limits based on making this distinction between industrial impacted limits and residential. So they set a limit of 125 parts per billion for if you're above that, you can't land apply. If you're below 50, you can land apply unrestricted. And in between, there are some restrictions that you can put limited amounts down. So, Bloom and the typical municipal biosolids easily meets that. As I mentioned, for PFAS, we're at 15 parts per billion, and their limit is 50. So again, some science went into this decision, and it still allows us to return this carbon and nutrients back to the earth from which it came. So this is really just reiterating what I just said. Maine set these limits based on these farms that had industrial sludges land applied to them. They painted all biosolids with the same brush, and they set these limits very low, such that nobody could continue to recycle this material back to the land. And Michigan made the distinction between the two types of biosolids. And again, our Bloom, which is derived from municipal biosolids, are above that main limit, but well below the Michigan limits. So with that, I could go on and on. Those of you who know me know that I'm very passionate about this issue, but I want to introduce Dr. Ian Pepper here. He did this study in Arizona, and he is continuing this work across the nation. We're looking for sites for this work. Dr. Ian Pepper, and yes, his name is Dr. Pepper. I'm going to get that right out of the way right away. Dr. Pepper is the director of Water and Energy Sustainable Technology Center at the University of Arizona, and is an environmental microbiologist. He's done research and has focused on the fate and transport of pathogens in air, water, soils, and municipal wastes. More recently, he has investigated the potential for real-time detection of contaminants in water distribution systems, some of this work that he's done here. Dr. Pepper is the professor in the Community Environment and Policy Department at the University of Arizona College of Public Health, as well as a professor in the Department of Ag and Biosystems Engineering. Many more great things to say about Ian, but I'm going to hand it over to him. Thanks.
Okay.
While Dr. Pepper is getting his slides up, just a reminder to type in any questions you may have for either speaker using the Q&A. Thanks.
Do I need to flip it, April?
You do.
Yep.
How's that?
Looks great. Thanks.
Okay. So, hello everyone, and greetings from sunny Tucson, Arizona. And the topic we're going to address today is looking at the question, is land application of biosolids a significant source of human PFAS exposure, specifically through groundwater contamination? So, a little bit on biosolids and land application. They're federally regulated by the Part 503 regs since 1993. We can designate biosolids as Class A or Class B. We've got about 7 million dry tons produced annually in the US, and about 60% of that is actually land applied. And you can see there a little bit of breakdown on Class A and Class B. And for sure, the major current issue is PFAS. And I've been working with biosolids and land application for many years, and it's interesting how the issues have changed over the years. So decades ago, metals were the big issues. Pathogens have always been an issue, particularly viruses, but PFAS is definitely getting people's attention at the moment. So these are poly and perfluorinated compounds. PFAS, they used to be known as PFCs. They impart resistance to heat, oil stains, grease, and water. And the two most famous, or perhaps infamous, compounds are PFAS and PFOA. And these are regarded by EPA as emerging contaminants. So, as Chris indicated, there is a lot of different household exposures to PFCs or PFAS, and in particular carpets, dust, baking and sandwich papers are some of the more predominant bouts of exposure. And there you can see at the bottom that PFAS was voluntarily phased out of production in the US early in the 2000s, and PFOA a little later. So why are we concerned about PFAS? Well, they're very persistent in the environment. They're resistant to microbial degradation. And the reason for that is the very strong bonds between carbon and fluorides. So this prevents their degradation. You can find PFAS worldwide in soil, sediments, water. And interestingly, you can find them just about anywhere. You can go to the Arctic and the Antarctic and you'll find PFAS. So you'll find them in places where there's no obvious source of anthropogenic activity. They are in fact ubiquitous. They're also soluble. They can migrate through soils. And it's thought that all people in the US, including me, you, everyone listening, everyone has PFAS in their blood at parts per billion. And if you're 40, 50 years old, you've probably been exposed for 40 to 50 years. That's the bad news. The good news is you can see here a little bit of data on blood levels of some PFAS constituents from 1999 to 2014. I don't have a graph with the more recent data, but you can see the drop there in PFAS and PFOA, and presumably that is due to the production being phased out. But of course, there are still legacy products out there that are still providing exposure. So EPA does regard them as emerging contaminants. They issued health advisory levels for specifically PFAS and PFOA. This used to be a combined 600 parts per trillion in 2009. 2016, it was reduced to a combined 70 parts per trillion. And then stunningly, in June 2022, the health advisory levels for drinking water were revised again downwards to 0.02 parts per trillion for PFAS and 0.004 parts per trillion for PFOA. And to put that in context, 1 part per trillion is equivalent to 1 second in 31,700 years. Respect to PFAS and biosolids, what is the exposure from biosolids? Well, the direct exposure is fairly minimal. If you're a responsible parent, you don't let your son or daughter play in a field which has just been land applied. And so the concern then is the indirect exposure, specifically the potential for drinking water that's being contaminated by leaching from the biosolids into the groundwater, or plant uptake of PFAS from land applied plots. Bioaccumulation is also a concern. So with respect to PFAS and biosolids, the issue really came to light back in 2011, land application in Decatur, Alabama, where biosolids, which had been contaminated by effluent from industries like 3M, were applied to agricultural fields. The surface and groundwaters became contaminated with PFOA at levels above the health advisory levels. And this began the scrutiny of PFAS in biosolids, which has continued and intensified over the years. There have been several studies on looking at PFAS in biosolids, but unfortunately, they tend to have some issues with the way these studies were conducted. Here's one looking at bioaccumulation of PFCs by earthworms. And this was a lab study. They took soil contaminated with PFAS, put that soil in a 1 liter Nalgene bottle. They then added 5 earthworms to each bottle in contact then with the industrially contaminated biosolids, very high levels of PFAS in the soil, and incubated them for 28 days. And of course, they found that the earthworms took up PFAS. But the real question is this realistic? A 1 liter bottle with industrially contaminated biosolids. Here's another one looking at uptake of PFAS into edible crops. Again, the soil in the greenhouse studies, the soil was contaminated with PFAS, so they were industrially contaminated biosolids. The biosolids were applied at 10 times the agronomic rate, and it was a pot study. So they grew lettuce and showed PFAS uptake, but the PFAS was spiked into the soil, and there are issues there which I'll highlight in a moment. In the field studies, again, both municipal and industrial biosolids applied at 10 times the agronomic rate, so high levels of PFAS. But even then, the uptake in corn was below the level of detection. And so the author acknowledged that land application is unlikely to be a primary source of PFC exposure. So the classic research mistakes, research mistake number 1, hot studies instead of field studies. If you're going to look at uptake of trace organics or metals, you should really look at field studies. You will get unrealistic data from pot studies. And we've known this since back in the 1970s, when many, many studies were done with metal uptake studies. Second mistake people make, 10 times the agronomic rate is not the same as 10 years at the 1x rate. You're really loading the dice when you do a 10x agronomic rate. And the third one, spike chemicals do not behave the same as chemicals that have been within the biosolids or end up in the biosolids following wastewater treatment. That being said, concern over PFAS in biosolids and land application, as Chris mentioned, led to a moratorium on land application in Pima County, Arizona, Tucson, where I live. This raised the cost of disposal or recycling of the biosolids from 1.3 million to 3.3 million annually. And of course, this got the attention of Pima County wastewater. And there have also been bans now in Maine, unprecedented. And I'm not quite sure what they're doing with the biosolids, because I don't think the legislators that banned the procedure had a plan as to what to do. So that's interesting. In response to the moratorium, we initiated a collaborative study between University of Arizona and Pima County wastewater. So this was a field study, March 2020. We took surface and depth soil samples from agricultural plots that had received known loadings of biosolids since 1984. So this was really, really helpful, a gold mine of potential data. We took soil samples, we analyzed for PFAS, also looked at groundwater samples, we had the appropriate controls. So pretty thorough study. This is a busy slide, but it's an important slide, so let me walk you through it. If you look at the field type there, we've got undisturbed soil, agricultural soil, and then group 1, 2, and 3. So the undisturbed soil is actually virgin desert, no agriculture, no irrigation, no biosolids. The agricultural field type is agriculture, irrigation, but no biosolids. And then the group 1, 2, and 3 are agricultural plots, irrigated, but with different rates of biosolids being applied cumulatively over the years. So this is a really fantastic opportunity to look at the incidence of PFAS in the soil following this land application schedule. We also were aware that you have to be very, very careful when you're doing PFAS analysis. You have to prevent contamination from extraneous sources of PFAS, because as I mentioned, PFAS is everywhere. So you can see all kinds of field clothing, cosmetics, sunscreen that you cannot wear when you're sampling, and you've got to be careful about what you wear. So this is a little intimidating at first. And again, with the sampling equipment, you can't use anything with Teflon. You can't use anything with low density polyethylene, a number of other items that you cannot use. And on the right, you can see the acceptable items. So the point of me showing you this is that, one, you do need to be careful when you're doing PFAS studies, and two, we were careful. As in a further example, I'll take you through the blanks that you need to check for contamination. And these are pretty strenuous. So the travel blank, you fill a 500 milliliter container with PFAS-free DI water, you cap it, you take it out into the field, you don't open it, you return the container to the lab, and then you analyze for PFAS, even though you didn't even open the container. The field blank is a different one. This time you fill up a container with PFAS-free DI water, and then out in the field, you transfer the water from that container into an empty container and return that second container back to the lab for PFAS analysis. And the third one is the equipment blank. And here, after decontaminating equipment in the field, you rinse what you hope is a clean auger with PFAS-free DI water, collect the rinsate, and return that to the lab for PFAS analysis. So when you first hear about these blanks that you have to have, it's a little intimidating. But take it one step at a time, and you can take care of the issues. So this is some data showing PFAS concentrations in groundwater used for irrigation. So you can see the PFAS analytes on the left, the contaminant, and then the agricultural sites. These are different wells used for irrigation of the agricultural plots with no biosolids. And then the group 1, 2, and 3, you can see these are the irrigation wells used for the various land application plots. And the important thing there is you can see in red that we are picking up PFAS and PFOA. So this is important, as we discuss in a moment. So now if you look at, this is PFAS in irrigated agriculture with no biosolids, and yet we're picking up PFAS analytes, and you can see the concentrations there of PFAS 1.85 at 1 foot, decreasing to 0.59 at 3 feet and 0.25 at 6 feet. So you can see the corresponding attenuation at the bottom of the data there, showing how much of the compounds are being removed within those surface soils. Also note that the PFAS analyte were present in the irrigation wells where it showed up in the soil. This is the low rate of biosolid application, and you'll notice the values there at 1 foot, 1.58 are actually very similar to the value that we had for agricultural plots without land application. And again, you see the attenuation depth from the surface of the soil. This is the intermediate rate of biosolids, 21 to 30 tons. You can see we've gone from 1.58 to 3.11 in the surface foot for PFAS, but these are still very, very low values. And again, you see the attenuation, and also you see that for PFAS and PFOA, this was present in the water from the irrigation wells. And here's the final one, the high rate of biosolid application. And now you see we're up to 4 parts per trillion, parts per billion. But again, very low amounts, and again, attenuation within that surface 6 feet. So you can look then at the mobility of PFAS by plotting the concentrations at the 1 foot, 3 feet, and 6 feet depths. And you see a rapid decrease in the concentrations, 2 orders of magnitude within that surface 6 feet. So this is important data. What are the highlights? One, low incidence of PFAS in the soils following long-term land application. PFAS soil concentrations in the agricultural plots that were irrigated were fairly similar with or without land application of biosolids, implying that both the biosolids and the irrigation water were both sources of PFAS. And importantly, 72% attenuation of all PFAS analytes occurred within the surface 6 feet of soil. So we presented this data to the Pima County Board of Supervisors, and this led to the moratorium on land application being rescinded in November 2020. So this was exciting, science-based regulations, what a concept. The other thing that occurred to me at that time was that we'd had a local problem in Pima County solved by a local study. And the thought occurred to me, well, then for a national problem, what do you need? And obviously you need a national study. Note there the citation for the peer review publication of the Pima County study. And again, this presentation is being recorded so you can get it. So we got the issue of PFAS and land application. And the big questions that raised concern nationally were, does land application result in significantly increased human exposure to PFAS? So that's an issue. And then will it lead to national or state bans or severe restrictions on land applications? And so again, the route of exposure we're concerned with, groundwater following potential leaching of PFAS through soil, and exposure to PFAS in crops following plant uptake. So this was led to the concept of a national collaborative study evaluating whether or not we had an issue with land application and PFAS. And the specific objectives that evolved for this study were to look at incidence in long-term land application at various soil depths, mobility by taking depth soil samples, and later on, critical to this was looking at the concentrations of PFAS in groundwater so that we create paired data sets. What's the concentration of PFAS in the soil? What's the concentration of PFAS in the groundwater? A similar concept with crop uptake, this time creating paired data sets of soil PFAS concentrations versus plant PFAS concentrations. Importantly, this study, we wanted to provide robust field data to allow for modeling that predicts PFAS leaching and the amount of PFAS that ends up in groundwater. And we'd like to use the model that's developed for site-specific evaluation of land application plots with respect to PFAS. Do specific land application sites are some more susceptible to leaching than others? So there are some unique aspects to this national collaborative project, and I'd like to point out how it's different from EPA-funded research on PFAS. First of all, it truly will be or is nationwide in scope, including studying a variety of different soils, depths to groundwater, different climates, and looking at land application across the entire United States, with looking at both irrigated and non-irrigated soils. Second important point is that the research methodology at each site will be identical. And this will allow for direct comparison of the data and a national set of real-world field data, allowing for the model development and quantitative risk assessments at specific sites. Also, I want to point out that we're looking at municipal biosolids, not industrially contaminated. If you're land applying industrially contaminated biosolids, all bets are off. So we standardize the research by having all the PFAS analyses conducted by the same lab. Also, the sampling and analyses protocols are similar at all sites. So we're doing exactly the same thing at different sites across the country. And all the samples are being sent to the University of Arizona, where we process them and then hand them over to the Arizona Laboratory for Emerging Contaminants, ALEC. So again, year one, soil samples taken at 1, 3, and 6 feet, groundwater samples taken and analyzed, samples collected from across the US. Where do we get the sites? Farmers with long-term land application plots with records of biosolid loading rates, academic researchers with established long-term land application plots. And this is all part of the W4170 multi-state regional research group. And we're looking for at least 30 sample sites across broad geographic regions. And so here's a schematic, 3 plots at each site. At each plot, we take 3 cores, and we have 3 depth samples, 1 foot, 3 feet, and 6 feet. So that's 3 times 3 times 3, giving us 27 soil samples from each site. These are the PFAS analytes. They're not there for you to read. They are there to show you that there's a large number of them representing the major PFAS analyte groups. Additionally, my colleague, Dr. Brousseau, is evaluating PFAS transport through pristine soils, no biosolids. This is part of a Department of Defense grant, 1.3 million. And so we can look at evaluating the effects of biosolids on mobility. We can look at transport with and without biosolids. And this also helps in model development. In year 2, we will be looking at crop uptake. We're not there yet. We're focused at the moment on the soil studies. But for the crop uptake studies, all sites will grow the same crop, harvest the same portions of the plants, analyze for PFAS, and again, the paired data sets of soil and plant PFAS concentrations. Just one question. We didn't have a grant, so how do we fund it? So the answer was donations from parties interested in land application. I started giving presentations around the country, promoting this concept of a national study. And importantly and excitingly, there was tremendous interest in developing this project. The people that were interested are utilities that recycle its biosolids via land application. The nonprofit groups, CASA, California Association for Sanitation Agencies. So CASA, NACWA, NEBRA, MABWA, we've been in touch with all of these people. These groups are well connected with utilities. And then also the private sector companies that manage biosolids. And this includes companies like Synagro and Denali Water. So we suggested donations based on from the utilities on the amount of wastewater processed. And you can see there the sliding scale and also the suggested donations for nonprofits, consulting groups, and private sector groups. And it worked. We formed an advisory committee, a detailed scope of work, and we have $450,000 pledged to date. This is enough funding for 25 to 30 sites. We've done site selection. We have 23 sites already selected. To aid each site, we produced a video on how to sample soils for PFAS without contamination. It's excruciating detail, but it's important to show everything. It's 20 minutes long, which is long for video. And there's a video link there that you can access that video and look at it if you're interested. So these are a list of the funding agencies, including DC Water, a lot of big utilities in there, Loudoun Water, sanitation districts of Los Angeles, Orange County Sanitation, private sector groups. So just a tremendous response to the concept of this project. So fundraising is still ongoing. We still need to raise money for the crop uptake studies in the second year. We're still planning. We've done site selection. We completed the first 2 sites in Tucson towards the end of 2022. Soil sampling is continuing, and the first external site was scheduled to collect their samples earlier this week. And we're currently looking at getting samples from the southern states, California, Texas, South Carolina, and then when the weather warms up, we'll move further north. So modeling the data, my colleague Mark Brousseau and Grau Bo developed a screening level risk assessment model, peer-reviewed publication. They've evaluated the model in the lab. What we do need is field evaluation, and we'll use the national project to generate that data. And the idea then is that you can input data from specific sites and look at the amount of PFAS in the groundwater, validate this model, and we can then use the model to predict the extent of leaching at specific loading specific land application sites based on inputting data from those sites. So site-specific evaluation, and these are some of the special analyses that will be needed, particularly the solid surface area, metal oxide content, clay mineralogy. Each site is doing the labor pro bono, one-time collection of soil samples. Everything's being sent to the U of A, and any relevant soil or biosolid data will also be sent. What does each site get? Well, they get specific soil, water, and biosolid PFAS data for their land application site, co-authors of the final report. And this is the important point, participation in ensuring the future sustainability of land application of biosolids in the US. So these are the states where we have sites, and keeping the sites anonymous at this point in time, but you can see we're doing pretty well. We perhaps need some sites in the North Dakota, South Dakota region. Oh, we have another one in Arkansas. So pretty good coverage of the US. And I'll finish. This is a similar slide to one that Chris had earlier. But basically, if you look at the concentrations of PFAS in various things, biosolids on the bottom, 23,000 parts per trillion. It's a big number. If you convert it to parts per billion, it's 23 parts per billion. Look at the concentrations in lipstick, mascara, the carpet, and in particular food packaging. So, you know, I raised the question initially, is PFAS a significant source of human exposure to PFAS? Relative to some of these other entities, no, it isn't. If you want to really expose yourself to PFAS, just eat some popcorn packaged in a container. And there are studies that have shown that the blood concentrations of people that eat popcorn are significantly higher. So I think it's important to put things in perspective. The project is successfully underway. We'd love for your participation in terms of if anyone has a prospective site, or if anyone's interested in helping support the project financially, that would be great. And here's the contact info. And with that, I will conclude.
Thank you so much, Dr. Pepper, and really exciting to see this important work underway. We have a number of questions that have come in for both you and Chris. Chris, if you could unmute yourself too and put your camera back on, that would be great. The first one I'll ask for you, Chris. So is DC Water participating in Dr. Pepper's study, and if so, how?
We are. We have contributed funds to COG, or the Council of Governments, which is sort of a local group that coordinates government efforts here in Washington, D.C. for the utilities. And COG has contributed the required $25,000. So yes, we are participating. We would love to have a site locally and a farmer who would cooperate to allow for sampling. So this is a pitch for that as well.
Great. Another question for you. Can you provide your perspective on your confidence in the sampling and lab methodology for PFAS testing? How do you see this evolving?
Is this for me or for you?
Yes, for you.
Well, you know, I always, whenever I see the data, I am confident that it is accurate and real. You know, I see Ian's presentation. I realize I don't know enough about the sampling. I've never done this sampling, but it is true. I mean, there is so many ways to contaminate a sample that we have to be extremely careful. So I don't know. I have to assume that the data that comes in is real and accurate, and then, you know, treat it as such.
And let me just follow up on that by saying that the blanks that you saw are a really good check as to whether you're getting contamination. So that's an important part of it. And when we get soil samples from other sites, the first thing we do is we have them send us a sample of their DI water and we analyze that and make sure that it's PFAS free, because if it isn't PFAS free, obviously you're going to get contamination. So it's very intimidating when you are first exposed to all the criteria for sampling and analysis, but trust me, when you're out in the field, it becomes routine very, very quickly.
Dr. Pepper, several questions around the levels of PFAS in Pima County, if you could remind us of that.
In the soil, you remember in the agricultural plots, with or without biosolids, they were 1 to 4 parts per billion, which is really low, really low.
The question may have been, what were the levels in the biosolids? I don't know for sure.
Yeah, we took some samples of biosolids now, and of course, it's interesting to see what they are, and then they're also in the low parts per billion, but that doesn't help you with the concentrations that were in the biosolids 20, 30 years ago, when they were certainly higher when, you know, inputs of PFAS were higher.
Chris, question for you around why the PFAS results that you had presented in your slides were different than what other groups have published about bloom, specifically the Sierra Club results.
Yeah, I'm very curious about that too. There were 2 groups that recently went and got bags of cured bloom from stores and split them open and sent samples in for analysis. And they came in higher, not an order of magnitude higher, but maybe like twice as high as what we found in our fresh bloom. And the working theory is that, for me anyways, is that, you know, those are in plastic bags. And some of those bags have been sitting for 18 months as they're waiting to get put on a shelf. And plastic, one of the uses for PFAS is to strengthen plastic. And of course, if you have a soil bag that weighs 50 pounds, you want a very, very strong plastic. We don't want it tearing open when somebody's putting it in their trunk at the garden center. So it's possible that it's coming from the packaging. I don't know. We just sent a sample of our cured material in to the University of, or Purdue University, to Dr. Linda Lee for analysis. And that was cured material that never touched plastic. We don't have those results yet, but it'll be interesting to see whether or not it comes back and has levels similar to the levels that other groups found. If it doesn't, then it probably came from the plastic. If it does, then it maybe has something to do with our process. Either way, the results that those 2 groups both meet the Michigan limit, and that cured material in the bag could be land applied in Michigan.
Still,
And I endorse what Chris is saying. Again, it's really easy to contaminate the sample. When we get our soil samples, the first thing we do is very carefully take out a portion of that soil sample that is actually going to undergo the PFAS analysis. And you're very, very careful how you handle that soil. We also partition portions of the soil out for soil moisture content and for routine soil analysis and for the complex soil analyses. But the first thing we did every time was get the sample that was going to actually undergo PFAS analysis. By the way, we also are collecting samples and archiving them, because I think getting soil samples from all over the United States is just a fantastic opportunity.
One other thing on that issue is a very, very, very small amount of our material, less than 1%, ends up cured and bagged in stores. The vast majority of what gets land applied locally and sent to soil blenders is our fresh bloom, which, you know, we have the samples that show that the results are considerably lower. And again, everything meets those Michigan standards, but we're trying to get to the bottom of why that cured material is higher.
great. Dr. Pepper, for the Pima County study, which findings were the most influential in convincing the county to lift the ban?
I haven't asked them. I think it's probably the whole package of, 1, low incidence after all these years, and 2, the fact that it was attenuating so quickly, I think is And again, the situation in Arizona, our depth to groundwater is 180 feet. And if you look at the potential risk of groundwater contamination, there are a number of factors that are important. The first factor, and the most important one by far, is the biosolids itself. If it's industrially contaminated, again, you're going to be in trouble. And there are a lot of studies looking at industrially contaminated biosolids that are land applied. If it's municipal biosolids, you're probably going to be just fine. The other factors you need to look at are the depth groundwater and also the nature of the soil and bodosome. Is it fine textured? Is it coarse textured? Those are probably the three major issues that will determine whether groundwater contamination is an issue.
Another question for you is your take on why PFAS, why this is emerging as an issue now, and what you see as driving the health advisory limit setting at the EPA.
So, tough question. I think it's emerging as an issue because, again, it's believed that everyone in the States has PFAS in their blood. And let me be upfront. PFAS is an issue. And at the moment, it's incredibly difficult to see how you could prevent human exposure to PFAS because it's everywhere. And the question with respect to biosolids is, if you have 10 different sources of exposure to PFAS and one source is a really high level of exposure, then it dwarfs all the other exposures. And if you look again at, well, just eating popcorn, I think is a great example. That can cause serious harm. And household dust, which is, if you've got carpets, if you're worried about PFAS, the first thing I suggest you do is rip all the carpets out of your house, put in tiles. Household dust is really nasty stuff. And, I think it's also becoming more relevant now. There are more human health studies coming out. For a long time, there was controversy as to whether PFAS cause adverse human health effects. It's now believed that they do. Liver, thyroid activities are, and it's also a suspected carcinogen. That being said, I should also point out that there are issues with toxicology studies. One, we do most of the studies on mice and rats, and two, a lot of the studies are high dose, short-term exposure. And what you really want are low dose, long-term exposure. And we just don't have that. And I'm talking about, 20 to 30 years exposure. But with respect to water, I suspect the way to the future is one, trying to take PFAS out of wastewater so that it doesn't end up in the biosolids. And I think that will be something we're going to be focused on.
Hey, can I add something, April?
Sure.
Just in defense of popcorn. I love popcorn. I eat tons of popcorn, but I have an air popper and I use real butter and put real salt on, and it's delicious. So what Ian's talking about is those packets of microwave popcorn.
Yeah.
And it's, we're creatures of convenience. We don't want that weird fake butter goo that's in there to soak through and get in our pants. And the thing that they do to the paper to prevent that is that they saturate it with PFAS. And that is in direct contact with popcorn. You rip that open and you're exposing yourself to PFAS. So I've banned that from our house.
I don't like popcorn, Chris. So I'm not an expert on it.
Well, I love popcorn. It's not the popcorn, it's the packaging.
Great, good points. Not to throw the baby out with the bathwater, as it were. We're at time here, but if if you are both okay with it, to plow through a couple more questions here, because I know
Sure.
we do have a number more, and there are a lot of people who couldn't join live, so this will give us a chance to address some of these as well.
Sure.
Going back to the study, Dr. Pepper, there were several questions about the irrigation water, where the irrigated water came from and how it was irrigated.
Yeah, we wondered what the source of PFAS was. And in fact, the sites that we sampled were chosen deliberately so that they were far away from obvious sources of contamination, like airports, industry. And we honestly don't know where the PFAS is coming from, other than to yet again say, PFAS is ubiquitous. You will find it everywhere.
Question about how the biosolids were applied at the sites, and would, that means of application, that change the depth concentrations?
Over the years, the methods of application for Pima County have changed. And the reason I know that is they funded me annually for 20 years. So I ended up with a 20-year field study, which is kind of unique. And the biosolids are either injected. Sometimes they were sprayed, depending upon the solids content of the biosolids. Most of it was injected into the surface feet, because that also helps with potential odor problems.
Have you engaged in any land application detractors in your review panel?
I'm sorry, say that again?
Have you engaged any land application detractors in your review panel?
Yeah, and this goes back to actually doing work with bioaerosols, when the potential for aerosolization of viruses was a big issue. We did another national study and collected samples all over the country and presented seminars and engaged activists all over the country. And my take home from those experiences were, there are three groups of people out there in the community. There's the one group that says biosolids is perfectly safe, you can eat it, won't cause you any harm. Well, obviously that's not true. There's the other extreme group, which thinks that if you get within a mile of biosolids, you will die. And in the middle, you have the vast majority of people that, when they find out that there is information, data, and science-based studies looking at land application, they are okay with it.
Just a couple more quick questions here. What PFAS compounds were analyzed in your Pima study, and how is PFAS in soils attenuated?
Again, 20 to 25 different PFAS analytes, of course, PFOS, PFOA, PFNA, PFHXS, those are the major ones. And so again, compounds representative of the different classes of PFAS analytes. And then the second question was, why is it attenuated in soil?
How?
It's mostly absorption at soil-air interface. And Dr. Brousseau is the expert on PFAS transport. Fine textured soils will immobilize more PFAS analytes than coarse textured.
And one last question, because I want to respect everyone's time here. We are running a little over. If biosolids were incinerated instead of being land applied, would that create a new pathway for human exposure via gases emitted? if you
I'm not a fan of exposure, period. Usually when you incinerate material, you end up with air pollution. And one of the problems we have, I teach a class called pollution science. And one of the things I point out is sometimes we don't really fix a pollution problem. We just move the pollutant from one place to the next.
There is no way. That's what my middle school science teacher used to say, and that stuck with me. Well,
I mean,
on that-
I think that that's maybe an explanation as to why it shows up in the Arctic is through atmospheric deposition, right? You know, we create a lot of trash in this country. That trash contains a lot of plastic, and what plastic doesn't end up in the landfill, or what trash that doesn't end up in the landfill, it ends up in an incinerator, and that goes right up the stack and out into the atmosphere. It's why it's in precipitation and rain. It's why we can find it everywhere, I think. So it's not a great solution, is my answer.
well, thank you so much, Chris and Dr. Pepper, for your time today. This was great. Thank you for everyone for attending. We will be sharing out these slides and the recording momentarily. So just look out for that. And thanks again. If you have any follow-up questions, obviously we couldn't get to all of them. We had over 300 people attend today. You can always reach out to Bloom at bloomsoil.com. Thank you so much.